Hi, everybody, and welcome to the Kidney Coach YouTube channel. I'm naturopathic Dr. Fiona Chin, and I'm really excited about the conversation that I'm about to have with you all today because I'm joined by two very esteemed guests. I'm joined with Dr. Alex Grunberg, who we've had on before. And if you haven't listened to the previous podcast about longevity medicine and all the amazing things that are coming down the pipeline, I highly suggest you go back and listen to that podcast. But we're also joined today by the president of something called Neo7, which we're going to talk about in a lot more detail further in the podcast. And the amazing John Catazzaro is here with us today. So I'll introduce both of our guests, but what we're going to cover today is all the latest information about new treatments and alternatives when it comes to working with a cancer diagnosis. And the big C is super scary for anyone to receive. But when you listen to these two gentlemen talk about the options and things that you may not have heard about, I really am hoping that by the end of it, people are feeling more empowered about the potential choices that you have when it comes to making decisions around your health and how to navigate a cancer diagnosis. So let me introduce Dr. John. So John is the CEO and founder of Neo7, which we will talk about further in the podcast. He's a doctor of naturopathic medicine, which he graduated in 1995. He's a doctor of theology and ethics, which is something always fabulous to have when you're a CEO of a big tech company. He's got 25 years of clinical experience, and he founded a cancer center and research institute outside of Seattle in Washington State. He's excelled in genetically matched immunopeptides, and he has a patented PBIMA platform conventor. John, I'm going to let you explain what that is later on. His mastery spans into millimolecular sequencing, peptide engineering, drug discovery, 3D proteomic integration, and it establishes him as a prominent thought leader in molecular biosciences. Half of those words I probably pronounced incorrectly, John, and I am so sorry. But his technological proficiency is rooted in aerospace roles. So, holy cow, he's worked for Boeing. He's beyond precision and individualized medicine, and he's got a keen interest in molecular peptides and engineering. So, wow, that is quite a big list of good credentials there, John. So we'll definitely get to more of that. And then Dr. Grunberg, who is also equally esteemed, he started his medical degree when he was 16, which to me is it leaves traces of probably a savant sitting there. Trained in Moscow, he completed his PhD program in clinical immunology. He's got over 3 decades of clinical experience in medicine, in immunology, biotechnology, longevity sciences. He also has a psychiatric PhD degree, which he doesn't often talk about, but means that he really understands the human mind as well when he's working with people, especially with big diagnoses such as cancer. He's worked in drug development, immune therapies, cancer research. He's an amazing practitioner. I often refer quite hard cases, especially cancer cases, across to him. He has an absolute fascination and medical knowledge in longevity medicine, integrative medicine, science-based approaches to promote healthy aging and prevent age-related diseases. He's worked in exploring breakthrough interventions such as gene therapy, peptide therapy, cell therapy, rejuvenation biotechnologies, all aimed at extending the human lifespan. He has held key positions, including medical director at Ivy and Rose, physician at St. Luke's Hospital, and resident at Mount Sinai. He's president of the GMD Bioadvance. He's led the company in drug development, successfully bringing multiple candidates through preclinical and clinical stages. And he's collaborated closely with the US Department of Defense to develop a program countering bioterrorism threats. Holy cow, you are both highly, highly credentialed, and I'm really excited to talk to you both. But what I want to start this topic leading by is just cancer and the state of where we're at when it comes to patients being diagnosed with cancer. And my dad died of cancer 2 years ago, so I know this very, very well. And I'll say it, you two don't have to say it, but the narrow-minded, very small window of treatment that patients are given, there is such a, in my opinion, lack of bravery in trying new frontiers when it comes to dealing with cancer because of how much fear there is around this word. And it means, I feel it really limits patients in the potentials that are out there. And I've seen what's out there and I see what you two are doing. So, Dr. Grimberg, I'd really love to hear about what you're seeing in the clinical world, because you treat far more cancer patients than I do, what's happening in the diagnosis and the process currently that patients are going through, and just how limited that is from your clinical experience.
Thank you, Fiona. Hi, everyone. And Fiona, thank you for bringing up this issue, this topic, because it's enormous, huge issue that clinical world, patients, everyone is dealing with, and working with patients and their families. I see that practically any family, any group of people has someone who is struggling with cancer, or their friends or loved ones, and this issue is involving everyone. And I have a great deal of compassion and respect for people struggling with cancer, as being privileged to have knowledge in both immunology and mental health. I see different aspects of people and their families being diagnosed with cancer and struggling with cancer, and their problems and difficulties in making decisions about their treatment, and not having enough tools to make educated or most logical and practical treatments. John, thank you so much for joining us. We will talk about this later, but I want to give good news from the start. So we do have more than most people know about treatment and successful treatment of cancers and other advanced diseases. And that's why John is here as a scientist. He will be able to explain more of it and give more details. But I will talk more about clinical issues and scientific and psychological aspects of decision-making by patients struggling with cancer and by their families. So can I continue?
Yes. Absolutely. I want you to really take us through. What I'd love you to do is and it's 1 in 2 people, it's 50% of people in our lifetime that are going to end up with cancer, I think, are the latest stats. And please correct me if I'm wrong there. Does that sound right? It's 1 in 2 people or something crazy. Yeah.
That's on point pretty much. Yep.
Which is scary. So we're all going to know someone. So Dr. Grimberg, what are you seeing? What's the current state of affairs at the moment with just the typical Western model when it comes to treating cancer? What options do people think they have, and how is that looking?
Let's start from diagnosis, because everything starts with diagnosis. So first, I will be talking fast for the sake of our audience time. So there are more than conventional diagnosis. There are certain labs, Grail and Gallery lab, that tests patients' blood and determines between 50 and 100 different types of cancer in very potentially at the very early stage. So it's not just this company, probably a lot of different companies now competing in this area for early diagnostic of cancers. So basically, for prevention of cancers, I would recommend taking this kind of test and having reliable genetic testing to see predispositions. That would be very important to increase awareness of potential cancer, and to, if possible, to change a lifestyle or approach to nutrition as early as possible to prevent certain kind of cancer. So this is a stage of diagnosis. And of course, I will not discuss all conventional ways of diagnosis, timely imaging, testing, or colonoscopy, or some other invasive tests to rule out cancer. It's very well-known information, but people should know that there are additional ways to diagnose cancer early, and it's always good to do so. So next, for cancers diagnosed at the early stage, or relatively early stage, actually, conventional oncology also gradually extends this term. So I have to praise conventional oncology that made tremendous progress the last few decades. And every year we see few new chemotherapy drugs on the market. We see more forms of immunotherapy. And in my clinical practice, I see survivals of cancer who struggled with cancer more than 25 or 30 years ago, 10 and 20 years ago. So basically, they achieved either prolonged remission or they cured, and that's wonderful to see. But there is a huge group of people. There is huge group of people who are diagnosed so-called late stage of cancer, and conventional oncology is frequently gives them the certain verdict and gives them very limited time to live, and that's very sad and painful. And in these stages, people have minimal choices as they think. They either turn to conventional therapy, combining chemotherapy and radiation, knowing that there is a high risk of side effects and relatively not high risk of curing the condition and significantly extending lifespan. Other group of patients, out of desperation and distrust to conventional medicine, they turn another way and they take different forms of naturopathic and holistic treatment. And I have to positively comment on those as well. I also see patients who are long-term survival of cancer and who feel that this holistic treatment gave them tremendous help in struggling with cancer. And of course, there are also miraculous but still existing cases when people get totally cured, even not taking anything. But again, all these cases on each side of this good outcome is statistically low. And I don't want to undermine all these treatments and decisions, but I just want to educate patients about more options that have a better statistical background, that are supported by solid and advanced science, and that may have ongoing due diligence or checking of, let's say, how do you measure if some modality of treatment helps? Probably the best measurement would be ongoing checking for cancer markers, circulating cancer markers, or cancer stem cells, or some other omics results of cancer existence in the body. John will tell more about this. So if we can do more ongoing testing, we can give reliable feedback if certain modality of treatment really works. That's very important. So, I don't think any facility in conventional oncology or in naturopathic world does this. I mean, in conventional oncology, there are certain types of cancer for which it's possible, but routinely it's not done. And patients don't get reliable feedback on how the treatment helps them. They just have to get like 8 or 10 treatments of chemotherapy, just hoping that it will work. But there is no interim progress report, no validation of results. Right, John?
Correct. Yes.
Yeah, it really sounds to me, sorry to interrupt you, like it's a guessing game. It's like, here's what we've got. We don't know if it's going to work. Here are the statistics of what it might work in this type of blood cancer or this type of cancer. Good luck. 8 rounds. There's lots of side effects, and we'll check in at the end to see if it worked. And if not, yeah, you might have an extra couple of months to live, or hey, we may have extended your life. But it's a complete guess in a sense, right? Of just based off best practice medicine from the past.
Mostly like this. Again, as in certain types of cancer, hematological oncologists, there are better markers, indexes that they can determine, like reducing certain kind of immature lymphocytes or things like this. But routinely for solid cancers particularly, I never seen any ongoing follow-up. And another issue I wanted to mention also, in conventional oncology, there is little consideration of coordinating time. I frequently see patients get the colon cancer removed and scheduled to get follow-up chemotherapy or radiation in 3 months. While logically it appears that it would be great to use radiation knife or chemotherapy agents to stop proliferation of cancer cells as soon as possible to the moment of surgical removal, if not simultaneously. But due to logistics of hospitals, of just system as a system, no one cares about this. So it's another huge problem. So basically, conventional oncology works as a, I would say, big factory, and it's not very discrete and individualized, and that's a problem. Definitely, oncology has to be highly individualized and customized, because every day I get calls from patients who get different effects from the same kind of chemotherapy, different effects, different severity. So it should be highly individualized, and this way, even in conventional oncology, I would hope to see better outcomes.
Yeah, I think that you hit on a great point there, Dr. Grinberg. I think one of the things that people need to realize that standard of care has been in motion for quite some time now, and standard of care is one size fits all versus moving to an individualized or personalized type of approach, right? So there's a gradual movement to go into a stratified approach, but it's very, very slow. And the reason why it's slow is because the way the system is set up, it's based on a payer system. So there's, different circles involved in this process and the business relationships, as well as, governmental concerns as well, because there's a lot of different grants and things like that in the research environment. And a lot of this is actually conditioned mainly by the academic institutions. And so the freedoms tend to be limited in choice. And in order for you to go to offer a better choice, you have to really be counterintuitive, and you have to color outside the lines, because of the fact that investigational means that if you're going to offer it to people as a personalized design, it doesn't follow a clinical trial schema. It doesn't follow any of the things that the standard conventional system has put in place. So it doesn't mean that we can't create the operative. We can, but creating the operative in a system that works very slowly and then hangs on to its standard of care as the first choice, and the first offerings, people then are limited, because they will not qualify for something that's more advanced and more hopeful until they first go through the standard of care options, which then wastes a lot of time. And of course, as cancer patients don't have time. And the grounds of experimentation either has to be very precise from the get-go. Otherwise, when you get into the actual progression of the illness, there's even a much more complicated scenario to work with now. And these are cases that we have picked up and that I've seen in my clinical practice when I was outside of Seattle. There we had 8 physicians, and we worked with a research team out of Dana-Farber, contracted labs there. And this is how we developed the whole process of being able to not just find the problem, but to come up with an engineered solution to the problem. Because that's the other thing that we face in this environment, is that everything is driven to finding, or diagnosing, which is important. But the thing is that if you don't have an actual solution engineering for what you find is a problem, that in itself is a problem. Because then you're telling patients that they have this diagnosis, and then you put a prognosis on that diagnosis, and then the patient is left, with their hands in the air saying, what other things can I do? And then they have to find, as you're probably familiar with, Fiona, as well, with your dad having the cancer, you have to start scrambling around to find out what other things can be done, because the you're not going to get that from your conventional doc. Your conventional specialist, your oncologist, is not going to necessarily, as Dr. Grinberg mentioned, you might have offerings for clinical trials, but these offerings for clinical trials, you don't know if you're going to get into a placebo group or an active group. And a lot of the times, again, you don't know what the ultimate turn. And then to qualify for these trials is another matter. A lot of the times they'll sign up for the trial. They go through the steps and they find out, maybe 2 months later that they can't be accepted into the trial. And that's 2 months more progression that a patient is going through, and because they were led by the nose by the system to try to hang on to a hopeful element that was not an option for them. And so anyway, all of these different things is the reason why Neo7 Bioscience is in existence today, because we've had many, many patients with very resistant cancer and with no hope scenarios, and told that they only had months to live. And that is exactly why we developed the technology we developed, because we wanted to demonstrate that we can make a difference in no hope scenarios. So, just so that we can point in the direction that you don't give up, you get better at doing the things that is going to offer a hopeful solution for patients. And to be individualized and specialized in individualization is as precise as you can get in understanding the actual relational value of the signals involved in the disease set. So in other words, it has its own whole signaling platform. You can have two patients with the same exact cancer diagnosis, but they may have the same gene target, but they wind up having a totally different engineered solution to that target based upon their profiling. So in other words, that's why, chemotherapies and other biological therapies and even traditional conventional immunotherapies tend to wear out their welcome, because of the fact that they're used on the mass population and they're not individualized. When you're tailoring something to the individualized molecular pathways involved that are faulty to the patient, that's as targeted as you can get for the patient. I mean, and then the other thing is that a lot of times conventional medicine goes after one thing. They'll make a cardinal doctrine out of one thing, whether it's a chemo that they've used for the last 3 decades, or whether it's a checkpoint inhibitor that they feel now can be applied to the more varieties of cancers. I mean, these are all good things, in development, but they wear out their welcome because they burn out, the people in the environment because they've used them or overused them, and they failed to use them in the right combination or the right dosing structure, or they've used them in combination with chemos that really have the opposite effects that you totally were planning on something working better, but the point is that they wind up pulling against each other instead of working together in a unified fashion. And thus the reason for more individualized targeting, because when you individualize target for these patients that have these resistant cancers, you are giving a chance for the other therapies to work much better. So in other words, it sensitizes the ability for conventional therapies to work more efficiently, because you are spending the time in isolating out the multi-targeted problem. So you're not just focusing on one target. You're not just focusing on PD-1. You're not just focusing on a LAG-3 or a CD27 or whatever the hot target is, like BRCA2 and some breast cancers, the BRCA family. You're focusing on a multi-targeted approach that's individualized to the patient.
And what I'm hearing, and Dr. Grinberg, I'd love you to talk about this, is essentially it boils down to fear, because we've got all these amazing options out there and people are given 1 month to 2 years, and oncologists that don't know about all this other stuff I don't know what it is, Dr. Grinberg, I'll let you talk about that, of why they're not looking into these more specific areas. But do you think it's I mean, because you've got psychiatric degrees in there as well, do you think it's a fear thing? Because cancer is so feared that it will, kill you. And we've then got 50% of the population going on to get cancer. And then are people hanging on to these old treatments because they are so scared to try something new in case it doesn't work? Are people so scared to look outside or even ask the questions? Because I know that was my dad, right? And he's into alternative medicine. He's got a daughter who's a naturopathic doctor who was saying, let's try all these other things. And he said, I'm going to do what the oncologists tell me to do, which is very common in a lot of my patients. And how do you see that play out in your patients, Dr. Grinberg? Because I think this is a big issue and maybe a blocker of why people are so afraid to look into these amazing technologies and treatments like Neo7 that's on offer.
Thank you, Fiona, for asking this question, and thank you, John, for introducing more about a scientific platform. Based on that, Neo7 helps cancer patients as well as other tough to treat and difficult to treat by conventional medicine diseases. Thank you. Yes, people diagnosed with advanced stages of cancer feel desperate, scared, lonely, and frequently their decision-making power is significantly less reduced, and their ability to notice, differentiate, and make decisions based on factual information is also reduced. Frequently, they delegate decision making to their family members. And here I see few different ways patients go. Like one way is just to follow conventional medicine because people are overwhelmed by authority and legality of their conventional oncologist. Other way is just try everything naturopathic, natural, and also because of deep-sitting belief and concept that naturopathic treatment is always better than, let's say, synthetic molecular therapeutics treatment. And I see other things happening as well. It's with very sad outcomes. There are some groups that unfortunately abuse people fear and desperation, and people go to different locations in different countries where they promise some kind of advanced genetic vaccine, advanced nucleotide vaccine, or some other fantastic methods of treating cancer. And while these countries don't even have infrastructure for basic molecular biology. And unfortunately, patients who don't have analytics with knowledge in biology, biotechnology, advising them, they just get kind of distracted and pay huge amount of money for these treatments. Like example, someone told me that she got treatment with proliferated NK cells, and I asked the patient how many days between blood draw and infusion of allegedly proliferated NK cells did you have? 3 days. And that's if people have a knowledge of biology, they know that the division cycle of NK lymphocytes, NK cells is 23 days. So if someone reinjects them after 3 days, it means it's just your blood plasma or whatever blood product is back, but it's not proliferated NK cell. And it's still they got charged huge amount of money for this. And for then when people see things like necrosis in the middle of the tumor and they ask these doctors what happens, they tell them that's a good sign. That means that the intervention is working, while any conventional oncologist would be very concerned by this grave sign. So, and unfortunately, people with advanced cancer are very vulnerable and easily manipulated in some cases. And another issue, in some cases, people delegate decision-making to their family members. The mental health professional also see that it's counterproductive because such life and death decision in such critical moments should be totally based on patient's knowledge, intuition, heart feelings, gut feelings, as family members can have more fear because of taking this responsibility in their hands, and they may not be so easy to make best decision because they are influenced by fear and burden of responsibility. For this reason, I strongly advise patients struggling with cancer to educate themselves as much as possible, as much as the energy and body allows them, and to make very educated decisions based on their feelings. And after all this being said, most, I would say more than 99% of patients struggling with cancer don't know that there are individualized and highly customized treatments that combine best features that I consider ideal for helping someone to struggle and overcome and even cure the cancer. There is reliability, there is transparency. I mean, the approach is totally transparent. They eager to discuss it with treating oncologist, with family member, explain A to Z of the treatment. Then there is a way to follow up during the treatment to see, for example, please correct me if I'm wrong, John. So to see traces of cancer cells DNA and RNA during the treatment, and possibly see a reduction of the amount, correct?
Yeah, this is exactly what we do on the analysis side, is we're looking at all of the patient's molecular target data, the bio data, which looks at their DNA, looks at their RNA transcription and translation. It looks at their total proteome, which means that it's looking at every dynamic protein in their bodies that are related to normal function and disease function, right? So we get to see the whole thing. And then, of course, the driving factor is matching their HLA immunophenotype. What that means is that we're looking at the central intelligence center of the immune system. The HLA is where we actually get our ability to design what's compatible to the patient. And we also get to see why the patient's resistance is occurring, because the HLA system originally was used to identify friend and foe, basically, used as a means for graft versus host, and to do tissue matching for organ donors, right? So the thing is that why did they use the HLA to do that? Because they want a perfect tissue match to make sure that they don't have a tissue that's going to be recognized by the body as an invader, very strongly, and then it winds up attacking, the transplanted organ, right? So the HLA system is used in our engineering to actually assist in identifying why the immune system is not recognizing the cancer. Why is it not finding the vulnerable places of the cancer? And it's very important as a driver of the design. So we find the bad targets, and then we create the central intelligence ability for recognition, for surveillance, and then we design the individualized peptides to those targets to either mitigate or potentiate or regulate, whatever it takes to create a better crosstalk ability to stop the cancer evolution, right? To stop that resistance pattern that the person's experiencing. And a lot of the times in cancer, in the early stages, it's easier to actually affect those signals more positively. As the cancer progresses, then you got more layered complications that you have to go through. It's not impossible, because we've worked with a lot of advanced stage 4 cancers, but the thing is that it's important to get things as early as you can. And but our platform is able to pick up on those very high resistance points. And that's really important, because in conventional therapies, a lot of the times when a patient's going through maybe 6 or 12 months of conventional therapy, they may be creating more resistance from the cancer. In other words, the cancer becomes more resistant, and it defies the laws of resolving the cancer, right? It actually creates more problems because the cells then become more immune to the person's own immune defense posture, and that's a problem because you don't want to be strengthening cancer proliferation by the therapies we choose. We want to actually make these cancer cells susceptible so that they can be attacked by those forces of the immune system, as Dr. Grinberg was mentioning, on the NK cell side of the house or the T cell side of the house. But even more so, how peptides work is they work by creating what we call fragment editing. So in other words, it's not just about signaling T cells, it's about changing the dynamic expression of the bad proteins that are manifesting bad signals, right?
So I'm going to put this into layman terms for people listening, because John is just this amazing brain. But what I'm hearing, John, please correct me, is the HLA system, basically, I always say it's like having Homer Simpson run your immune system versus having Elon Musk. So when we have a cancer proliferation, Homer Simpson's, he's got his donuts, he's at the wheel, things have gone awry, the body can no longer tell the difference between cancer cells or not. And as people mostly know, we all have a level of cancer cells. The immune system's clearing them, it's staying on top of them. What happens in a cancer proliferation is the immune system no longer recognizes them. Cancer cells are sneaky little suckers. They can coat themselves, change their polarity, do all these things to stop the immune system from attacking them. What it sounds like Neo7 is able to do is to go in and find out exactly why this HLA controllers, be it why Homer Simpson was hired versus Elon Musk, fire Homer Simpson, put Elon Musk back in the driver's seat. So the immune system goes, oh, hello, we've got cancer cells, and it can mop it up, meaning that then your own immune system, via using peptides, is able to enhance its natural killer cells, T cells, to get on top of there, which would not only then enhance your own immune system's ability to clear cancer cells, but any therapeutics that you may have picked up in the Neo7 that will then enhance that or work alongside versus against is going to have a better chance of working. Did I get all of that right?
That's perfect. I love the Homer Simpson versus Elon Musk approach, because that's actually a perfect analogy there, right? To kind of put together a perfect comparison, and it actually fits exactly with what we're trying to achieve. So bravo on you, Fiona. That's excellent.
And I also want to say what I'm hearing, and I don't want to get too sidetracked from cancer, but being someone who works a lot with autoimmune disease and having autoimmune disease myself, which I also know is a fault in the HLA genes, this is not just applicable to cancer. This is going to apply to anything where the immune system has become faulty. Is that right?
That's correct. So now, like in our combination product that we have, which actually looks at the complete what we call multiplex data sets that a person's body will manifest, right? So in other words, these data points, and let's take autoimmune as an example. We're measuring every transcription expression in cytokines, chemokines, regulation target cells for the immune system. So we get to see whether they are a hefty red or they're a favorable green. And if you're actually in the red, the red can go two ways. Either the red is because it's not protecting you enough, or the red is because it's causing a lot of trouble for you, right? In the immune system. And then we intelligently know how to toggle those switches. Then we're saying, okay, so this target needs to be potentiated because it's red, because it's underexpressed, and we need to bump up the margin. So I'll give you an example. Interleukin 10 is a master regulator of the inflammatory cycle. So if you have an underexpression of interleukin 10, you're going to have an altered expression of the control cells which are known as CD25s, and you're going to have an accelerated amount of inflammatory destruction because you can't regulate the interleukin 10. Well, what our design will do is potentiate interleukin 10 to allow interleukin 10 to become more sensitive to drop down the excessive inflammatory and target destructive activity, right? Because you can you can see relationships there. When you have a low expressed interleukin 10 and a very high expressed interleukin 6, or a very high expressed interleukin 6R, which is a receptor, or a bad toggled interleukin 19, these are all important players in the bigger picture. So if you are just focusing in on one target and only one target, and you miss the other ones for proper solutions, then you can definitely, impair your ability to really hit the problem to where it needs to be hit, right? So in other words, create the signal changes that need to be changed to create that balanced effect again, So autoimmunity is and then the other thing that comes into play there is when you identify the possible triggers that created the autoimmunity. So in cases of Lyme disease or viral activation or some kind of an environmental toxic hit of some sort, you get to see what the body's real-time measurement is. So in other words, you have the DNA. DNA is not always a good telltale. The RNA transcription is a very good telltale because it transcribes and translates. It's the typewriter that keeps on typing. So your typewriter keeps on typing. And if you get an error in your ability to construct the right wording, then boom, it goes back to the DNA over time. So in other words, the next cell cycle can bear the bad typing message over the next cell generation, right? And we see that with cancer. We see that with autoimmune disease. We see that with neurodegenerative disease and faulty protein activity. It's just amazing how the great reveal is in this signal platform.
So, and I'm going to throw back to you, Dr. Greenberg, but what I'm hearing is it's like, and Dr. Greenberg, I'll let you talk to this, but it's like our conventional medicine goes, okay, I'm going to use chemo and radiation. We know that works, but it's like having an orchestra and we're just going to take out the trombone player because maybe he's the problem. We're going to knock him out. He's slightly out of sound. And maybe that'll fix the problem. But actually, the violin and the flute aren't tuned properly. And what I'm hearing Neo7 does is we're looking at the entire symphony and we go, look, Mr. Violin player, you need to play a little faster. The flute, you're off pitch. We need to change you there. And you're looking at the entire symphony, how it works together so you can create proper music versus just wiping out the trombone and hoping that the orchestra's going to sound better.
Yes, it's a good point that you're making. It's kind of like when you first walk in and you're getting the tune-up in the orchestra and there's just all these dissonant sounds and bang, 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 and you get all of these stray, everybody's trying to find the right note and get on key and figure it out, and then all of a sudden, boom, they're all ready to go for the performance.
Yeah. So, yes, Dr. Greenberg, how do you apply this? How do you move patients across from where you're seeing them conventionally to here? Like, how do we bridge this? Like, what are we doing?
So please let me be a translator of scientific language into language I speak to patients with some kind of liaison for John and also be clinical liaison for you as I hope some other healthcare practitioners may be listening to this, and I can provide some kind of clinical information about this. So the point is that, for example, cancer has many manifestations in the body, not limited to its locality and microenvironment. So it means that struggling with cancer definitely affects how this person's immune system gets antigens represented, how they get processed, how the information gets transferred from different subpopulations of lymphocytes, and many other processes. And definitely there is certain level of changes in cytokines or inflammatory-related cytokines that is all involved in the process of cancer cells escape from standard chemotherapy, resistant to radiation, ability to mutate. These renegade cells that obviously think they are first to get immortal in the human body develop many mechanisms to escape toxins, to escape radiation, and to proliferate indefinitely and to be immortal and even to steal mitochondria from healthy cells. So all modalities of conventional medicine targets one point like that. And definitely even combining them doesn't target all signals across the body. People should realize that every cell communicates with every cell. They send each other emails, they send each other telegrams, and they make phone calls, and usually in the form of small molecules called peptides. That's the easiest form of communication. That's why a Neo7 platform for analyzing all these normal and faulty signals and analyzing them in context of considering HLA and doing certain matching using AI is a great tool to understand what happens in this enormous volume of communication between cells. And then next step is to find how to affect the signals to bring back the body from faulty signaling to improved and eventually signaling associated with no active disease or prolonged remission. That's what's happening. Yeah, and again, this signaling John was talking about, they mostly conducted by means of peptides being released, synthesized by cells, by other cells expressing receptors to be able to, like antennas, to be able to accept this transmission as it's supposed to be. So it's a very complex process. There are expression of genes inside the cells that regulate expression of receptors on their surfaces and make them accept or avoid receiving certain signals. So it's enormously complex process. And if you consider that it's not just one group of genes, but each genome uniquely has different groups of genes regulating each other, following their own very hidden, subtle mechanisms, then you really need to see real-time signaling and be able to affect it and get feedback, what happens, how you fix the signaling. Yes. And based on this, actually, there is big overlap between topics in oncology and longevity. And that's why Neo7 started using this platform also for longevity applications, because in longevity, as in other clinical disciplines, and of course in oncology, it's important to simultaneously and synchronously to affect expression or signaling in all systems. Otherwise, working only on one system, or like say in case of gene therapy, injecting only one gene, or in case of transfusion of young blood plasma having certain amount of ingredients with temporary effect. Quite an opposite. You need to work constantly and simultaneously on normalizing and eventually finding changes in expressions of genes and signaling associated with younger age. Then you can deliberately, at least at the epigenetic level, reprogram cells signaling back to younger songs. And that translates into younger sensations and obviously improving of hallmarks of aging as well.
Yes.
Now, I know a lot of our listeners would love to hear some case studies or some stories where you've had maybe a patient that was resistant to things that has gone through this process and just what's happened. And for people that haven't listened to any of our previous podcasts, you'll start to understand why I am right down the rabbit hole of longevity and peptides and biohacking, which is that movement is starting to move across into our mainstream medicine, which is what Dr. Grinberg is talking about, and the importance of peptides and their role on how it unravels chromatin and changes DNA expression and those sort of things. And I've said for the last 2 years when I discovered peptides is they are the future of our medicine. There's no doubt in my mind that that's where we're headed. They have basically no side effects. And when you get the right ones, and of course I spend thousands of dollars a month on peptides, I could have a Neo7 test, which would actually tell me the best way to optimize my genome. And I could cut all of that out. So I can start to see now how the Neo7 will apply not to just cancers and autoimmunity, but the biohacking field that are maybe just injecting BPC and some human growth analogs and some melanotans and whatever. If they had something like Neo7, they could actually have the map to optimize their genetic expression, which now we're talking what you're talking about, Dr. Grinberg, which is we're heading into longevity and some very exciting things. But I digress. I'd love to hear of some cases where maybe someone had cancer, traditional treatments weren't working, and how you were able to use something like Neo7 to improve their outcomes and what that looks like. I'd love to give some people some hope and some understanding of why maybe doing this testing, because I know it's not cheap, so we'll talk about that too, but why looking at something like this actually may be cheaper than the guesswork that a lot of people are doing.
Yes, this is a very good point. And also, we'll continue on this topic. And I wanted to add, considering main direction of your podcast, Fiona, that Neo7 platform, like John was talking about autoimmunity. It's a big, very important issue, autoimmunity and everything that happens in the immune system, different forms of activation of immune system. So now conventional medicine considers more than 100 diseases as having autoimmune cause, or at least significant contribution from autoimmunity. So it's extremely important area that's leading to mitochondrial attrition, to telomere shortening, to different forms of inflammation, inflammaging, neuroinflammation, translates into hundreds of different chronic clinical symptoms, premature aging, to so many things, and obviously contributes to probably to etiology of cancer as well, because immune system is distracted by unnecessary activities. So in kidney disease, that this is a main topic of your podcast. Kidney diseases, the inflammation and autoimmunity plays a very significant role in kidney diseases. So luckily, Neo7 also, when I discuss cases of my patients, I have different cases, autoimmune or different forms of kidney conditions, different forms of nephritis, and people with different with other autoimmune conditions. So I consult with John and his scientific team, and I hear that they can accept and help the patient. I would say most, if not all cases, rare cases and hard to treat cases that I have to refer. And I'm very thankful for this. Diabetes, huge part of diabetes is autoimmune causation, origin, and definitely reducing the production of antibodies attacking beta cells or insulin would be very huge help to people suffering from diabetes. If it can be affecting even deeper level of immune system, of transfer of information between memory cells, then it would be even more probably permanent and deeper solution to treat autoimmune diabetes and similar conditions. So, back to kidney diseases. In addition to a number of pathological kidney conditions that lead to chronic kidney disease, there are huge number of cancers arising from different tissues, from kidneys and urological system, and that's where Neo7 has also good expertise. And I enjoy studying cases when I refer patients. John most of the time has similar cases, even for rare tumors, some rare forms of sarcoma or glioblastoma multiforme. So it's a pleasure to consult with John and his team.
So tell me more, John. Yeah,
yes, if you want, I can share the screen. Share the screen briefly, if that helps.
Yes, let me give you. I need to give you the power to.
Yes, I need the power. Okay, so this is this is a real patient. Right. This patient actually has Alzheimer's, and we addressed his Alzheimer's by doing a primary design. But then he had a very layered diabetes, very strong diabetes, right, type 2, which actually was putting a very heavy load of obstructive activity on his kidney. So because the priority was to address his Alzheimer's, by the way, after 3 months worth of therapy on his personalized peptides, his Alzheimer's got like 75% better, to where he was actually going out and walking and even doing slight jog for 3 miles a day. This is a guy This is a guy who's in his 80s, by the way, not a young fella, a more seasoned fella, but he was definitely disturbed about his decline. And so within 3 months' time, we were getting some very positive results with his Alzheimer's. But then the doctor we're working with said, you know what? He's got a really aggravated situation going on with his diabetes. So what we did is we did a profiling where you can actually see his insulin signaling is terrible. It's in the tank. The expression's 3.6 down. He's got a lot of down regulated expressions, but a lot of these down regulated expressions can be toggled. Like, for instance, if you improve the insulin signaling and you improve the polypeptide development, and then you improve the actual CDK5 signaling, a lot of these switches will then, start to turn around in a favorable green, right? So you don't have to mitigate every one of these. You have to mitigate the ones that are the top targets. So the ones that came up the top as the top signaling troublemakers were the ones that you see in green that have to be potentiated to create better signaling. Okay. Then we move down to the actual glucose metabolism, and you can see that he's got a very hefty MTHFR right there. Whereas the methylation is quite off. But the one that actually creates the biggest metabolic burden is the DPP4. So the DPP4, when you actually mitigate this, then and then you do some other things to address the MTHFR, you can significantly change the profile there. And then we looked at the pancreatic beta cell signaling. So he actually had a WFS1, which was a very down-regulated signal. You can see that this one is really indicated in the endoplasmic reticulum signaling. You know that the ER is responsible for packaging up and getting the right transcription translation going. And if you have a dysfunctional ER, you got some significant problems with sensitizing the pancreatic activity. So the main one here that we saw as a big troublemaker was the WFS1. But then here's the real one here. UMOD is actually a regenerative target protein that is responsible for functional restoration of the kidneys, right? So he's got a functional down regulation on that transcription, which is creating more impaired kidney disturbance to where his kidneys are not, he's very close to being, going on dialysis. And you're going to start seeing UMOD changes like this whenever you start seeing more kidney destruction. So what we did is we have a design that in the design, we included a potentiation of the UMOD. And then, of course, now we get into the inflammation and immune regulation part. Whereas IL-7 is off, his IL-10 is off, his IL-15 is overexpressed. And I believe he's got, the main ones here, as you can see, is IL-10 is down expressed by 6.7. This is actually supposed to be expressed like in favorable 2.0 in the green for a positive, right? So, and then here, IL-7 is creating a lot of immune burden by being overexpressed. So that one is mitigated, right? So his design basically for the revision included some significant focal points, as you can see here. So we mitigated in the red these particular gene targets. We potentiated insulin and PPY targets. We potentiated UMOD target, and we mitigated on IL-7, and we potentiated IL-10, and we mitigated 15. Now, when you mitigate 15 and you mitigate 7, then the other inflammatory cytokines then toggle back into a better expression. And then at the same time, you're regulating interleukin-10, raising the bar for better regulation. And then we also put in there an anti-galectin-3 peptide complex personalized to the patient. So this is the second design for the patient that he's going to be starting soon to address those concerns, right? So you got there's a total of 4 sequences there that are directed toward cell signaling and correcting those error signals. And then the other thing I wanted to show you was with the longevity, really quick, is that we have a total of 36 patients in our longevity pilot. We have, and then we also have 5 normal patients that were volunteers. So totally, there's a total of 38 patients in the pilot. And so what we did is we focused in on what the human cell communication network is, right? So what Dr. Grinberg was referring to is that, you know, our cancer work and our work with other, you know, complicated disease sets, we started with cancer, and of course we've layered it on some complicated autoimmunity, neurodegenerative, and other metabolic disturbances, has taught us how to better navigate the longevity world, right? And the aging hallmark. So what we found is the most important center is the human cell communication network. And as you know, just in the basic science of it, is that we got what we call a pre-DNA structure, where you have the DNA transcription and then a pre-messenger RNA movement. And then as you can see, as you're going down to this whole process, it's simply transcription and translation. So everything that affects a protein in the protein signaling path that affects a lot of functional integers like, you know, interleukins, chemokines, T cell receptors, NK cells, all of these defense stem cells, and even cancer stem cells, right? All of these things start with moving into a faulty transcription translation relationship. And as you can see here, this is which points out the science of it. But one of the things that we saw that's a common denominator, because we measured 8 aging hallmarks to start in the pilot, but out of the 8 aging hallmarks, the 6 core impacts that we noticed in these patients is there's a common denominator between the inflammatory and oxidative burden, the mitochondrial dysfunction, the instability of the DNA RNA proteome, and the protein translation with aberrant signaling, and then the oncogenic burden increasing, and then telomere instability, right? So we see those 6 core functional impacts. Well, let me just show you here. This is like an example of a patient's, real-time patient's target genes that are involved in the different areas, specific impact areas. So as you can see, this patient has very impaired inflammatory oxidative adaptation. Their PARP1 is a main troublemaker. The SH2B3 is a main troublemaker. And then, of course, they've got reduced oxidative protection by having reduction in oxytocin, reduction in SOD3, and reduction in the LMNA, right? And so what that does is that affects the transcription rate and the DNA repair rate and all of the other things that are going to help reduce the risk potential. And what you'll find is you have increase in the risk potential in the inflammatory destruction, the oncogenic potential, and an adverse effect on telomere attrition, which you don't want, and then an instability in the RNA DNA proteome, okay? So now that is actually a window into a real patient. And then what we saw in this patient is we see the expressed relationships, like unfavorable expression, either too upregulated or too downregulated. So the minuses are downregulated, unfavorable, and the pluses are upregulated, unfavorable in the respective aging hallmarks, right?
Yeah.
So now the bigger picture, I mean, here's, if you want to talk about, you know, where signals and crosstalks and protein dynamics are, well, then you're starting to see a lot more of these affected targets that intercommunicate with each other, like Dr. Grinberg was saying, sending emails, having phone conversations, all of those things, which are very, very, very true when it comes to PPI, protein-to-protein interactions, right? Well, let me just show you one other thing. This is on a doctor that we work with who is a longevity doctor, and this is his histogram. And his histogram basically, I'll put it in presentation mode to see if you can see it better. Can you see that better now? Is that better? Can you see that?
Yeah, that's perfect.
Okay, so we looked at, here you go, looking at the key indicators and those aging hallmarks that we talked about. And in the beginning, where you see a lot of the red, up until you see where the arrow is, right? So there are some, like when he first started out, he was not too bad on autophagy and pretty much stayed in a good autophagy rhythm, but his longevity was kind of, starting out not so good. Then he made some improvements by doing things like EBU and plasma exchange and some other things, telomerase. You got a little bit of activity here with maybe budging longevity core gene sets, but his telomere attrition was getting worse, and his inflammation and oxidative stress was getting worse. So as you can see, he's got resistance in 1, 2, 3, 4, 5 interval follow-ups to where he was not budging the meter at all. So now the arrow is where he did his personalized peptides for 9 weeks. He did 9 weeks of personalized peptides, did another interval follow-up by sending another sample in. And then what you can see here is that he budged the meter on the longevity. He budged the meter a bit. He budged the meter a bit on the telomere attrition naturally. Improved, even got a better result on his autophagy. His inflammation and oxidative stress burden really improved. So he really broke the resistance there. And then we got him back in the green, back in DNA, RNA, proteome stability, right? And then the other factors with oncogenic potential was good, and his SAS burden was good. But now the really interesting phenomenon is that we had aggravated mitophagy. And what happens there is that when your body is creating a cell correct over new generations of cells, you have a higher mitophagy rate. So a lot of mitochondria that are bad, that are faulty, and that are limp along mitochondria, they go into a mitochondrial death, right? So it's like an apoptotic effect on the mitochondria. So that's part of the renewal cycle, showing that we're moving in a favorable direction, right? Now, he was doing a lot of different things here when you see these resistant reds. He was doing a lot of plasma exchange, plasmapheresis, EBU, a lot of enzymes that, specifically to activate telomere pathways. But a lot of the times what you wind up seeing, and I'll stop sharing, what you wind up seeing is you put the cart before the horse. Like, in other words, you think that if you're going to really make an influence on telomeres, that you need to really go direct on the telomeres. No. What you need to do is you have to correct the underlying stressors that create the telomere signaling disturbances so they can naturally cycle into a better chromosomal stability. So if you have a lot of inflammatory oxidative stress burden, you certainly don't want to be trying to elongate telomeres while you still have an underlying root issue, right? Bad signaling, bad mitochondrial signaling, bad inflammatory oxidative stress signaling. You got to work with these signals to really, and then a lot of these things naturally shift into a favorable rhythm. So each one of those histograms that you saw has a core gene set behind it in its real-time transcription that are measured by the platform. The platform measures these relationships, and then the output engineering specifically geared toward those particular targets that need the priority. They have to be triaged, right? So anyway.
And it makes sense too, because I'm also, for people watching, a small nudge in how your body deals with, say, autophagy or working with telomeres actually has an exponential effect on function. So even though people might be looking at the graph going, it didn't move much, know how that translates into health and the ability for, say, the kidneys to reverse things like fibrosis, or say someone with an autoimmune disease for HLA cells to actually see what's going on, or cancer cells with autophagy to be able to actually get on top of that sort of stuff. Those small bumps, especially when combined, actually end up in really big signal changes throughout the body, right?
Yes, that's so true. And the other thing is, when you have accessory function. So, like, in other words, if you have a problem with, say, a person they injured their hand and they're going through physical therapy to try to get strength back into their hand. Well, what you wind up having is this accessory functions that help with maybe you can't move this one digit because it hit the nerve there. But with over time in physical therapy, you'll have some accessory help where you have regenerative activity happening to where you do have that recruitment to allow for that function to happen. Well, I like to use that as a means to kind of like say the body is an amazing engineering, because it actually can create, if you have fibrosis, a lot of the times people think, oh, you got fibrosis, that's the end. We actually have patients that have pulmonary fibrosis that have a little bit of lung tissue that they can work with to create an accessory effect, to create function, better function, and maybe they're working on a third of their lung, but they have a capacity now that's increasing because they're regenerating the tissue that's there, that's vibrant and vital, right? So as long as your body has a substrate, a substance or a moiety to work with, the fibrosis is not the end of the world. You can change by creating an accessory response. So that's how the regenerative world works in the body. It creates that accessory response. It works over cycling over time and cell cycling and renewal. It's just an amazing way to view resilience.
What an amazing tool you've come up with, John, and I can see the engineer in it, and it's just beautiful how engineering and AI can then go back into these processes in the body and upregulate them and improve our health. So I really appreciate your brain and what you've brought to it. Dr. Grinberg, so my question now is to tie all of this up, because I think we've got a really good idea of how Neo7 works, and we'll get into that. But if you're a patient and you're diagnosed with cancer and you've just listened to this podcast and you think, hang on a minute, there's been a big gap in my healthcare here. There's potentially a test that I could have that will essentially, what I'm seeing, give me a roadmap on how to improve my autophagy and how to make my, mitochondria work more effectively so I can mop up cancer cells and get my HLA genes to switch on and reduce inflammation. Where to from a patient from here? How do they go from listening to this information to then working with a practitioner? Because I know there's not many of us around the world that have even heard of Neo7. How do we get brave enough with all your training with mental health and how the mind works to start to make these decisions to look at that? How do we move into this world?
Thank you. So first I wanted to explain a little bit of what John was showing before in the clinical and simple terms that patients can process and can turn into actionable knowledge. First, I want to emphasize the testing result that John showed initially. It's very interesting because many people I treat in integrative medicine, they're looking for different genetic tests, and I have hard time finding company that provides actionable knowledge, that in the reports provides more than just generic description. And here, John table had one more column, so it had RNA expression there, right? And that's what the tests are missing. And this RNA expression is very actionable knowledge, because based on this, Neo7 synthesizes certain molecules that can affect this RNA expression, correct? And we could see it in the table how the dynamic over time. So it's applicable for any condition, for cancer, for autoimmune disease, or in cases where people want to seek help in longevity, age-reversing medicine. So it's so important. That's so important for cancer patients because, particularly in cases when there is very limited time and limited options, it's so important to have actionable knowledge how to start treatment and how to monitor the treatment along the way in real time. It's so important to see that treatment helps, and then you can quantitatively and qualitatively modulate it to add something and to affect the cellular signaling. And it's interesting working with John and Neo7. There is also another company that specializes in transcriptomics called Selecta Bio, and we develop testing where we take blood from samples of blood from my patients and incubate them ex vivo with peptides or agents of interest to see in real time how it affects many thousands of genes, the expression. It's so very interesting. And we're talking about combining this approach to even add another level of accuracy and predictability to, if we can, before we start treatment, to incubate potential prospective patient blood sample with peptides, molecules synthesized by Neo7, and then we can, through qualitative and quantitative assays, see how to optimize the treatment. That would be kind of holy grail of treatment in this area, right? And the same stands for kidney disease. As you could see, John was explaining, demonstrating case of patients with mild Alzheimer, and also had diabetic, obviously, nephropathy and some autoimmune and inflammatory conditions. So, you could see that a lot of target genes were assessed. Like last one was something related to galactin. Some company use monoclonal antibodies against galactin as a monotreatment for certain conditions that works very well. At the same time, John showed a couple of dozen similar molecules that they affect. So you can see how much this treatment would be more efficient than just affecting one site, right? And John also showed the changes over the time in all these areas. And that's autophagy, mitophagy is very important process, how mitochondria clean themselves. They, when they get older, they just eat the aging parts and giving place for younger parts to come. And that's very important process to rejuvenate. And I'm very happy to see that Neo7 platform doesn't miss any important hallmarks or markers of aging, all this mitochondrial, subtle, intimate processes. And I'm sure there are more studies on more subtle process affecting telomeres and related genes that the signaling also affects how telomeres health maintains itself. So many things, each of them probably would be a topic for scientific work. So for patient, yeah. If so, in case that's what I do. I when I have patients with advanced cancer and strict and scary verdicts from conventional oncologists given, I refer them to Neo7. They get very good education there. The oncologists, the families, all welcome to join, and it's very transparent with all these explanations at the highest level. And that's compatible. These treatments are compatible with chemotherapy, and Neo7 also goes a long way to make to be able to create complementary treatment. So that's great. So I think that's great resource, and I'm very happy that we have a chance to educate people who may struggle with very tough, grave, or terminal illness, to find out that there are ways to help them, and they are very scientifically based, hard evidence based, and very advanced. And from clinical side and from statistics, we see higher success rate, higher than anyone else can offer for these conditions, and very low amount of and low intensity of side effects. Most of the times treatment's pretty well tolerated, and they consist from courses of IV infusions of the molecule synthesized by Neo7, then processed in special compounding pharmacies, and then being administered by research operating specialists or sites, physicians like me that Neo7 is collaborating with. So we're very happy to have this pattern of interaction, and to have Neo7 providing us with a means to help people.
So if a patient wants to get hold of Neo7, do they need to go through a practitioner like Dr. Grinberg first, John, or can they go straight to the website and contact you and say, hey, my oncologists are not open-minded, help, I want to do this test?
Well, going through Dr. Grinberg will accelerate the ability to find a practitioner, a specialist like himself who works in the program. But they can find more information on the website, the www.neo7bioscience.com. And find out more information there. And then if they're interested, then we wind up we call our first webcast or team meeting with the doctor and the doctor with the patient. And we have a collective, it's usually about a 45-minute, between 30 and 45-minute discussion about the whole process from cover to cover. And, explaining that all of the things that we work with in the development are QA, QM, every step of the way. Like, for instance, our contracted labs are very specialized labs. In other words, you can't, it's not just like sending a blood sample to go to Gallery and getting a ctDNA read. It's a lot more in depth than that. We're looking at what we call high definition reads, because remember, what we're doing is we're producing an output engineering. And when you're producing an output engineering, you can't have little array type panels that give you just scants of information and don't have the whole picture. So you wind up getting the entire picture with our contracted labs that multiplex this data from the standpoint of the proteome, the RNA transcriptome, and the functional part of the genome, and the HLA immunophenotyping. And then once they get educated on the process, they'll see that it's not like just going to a compounding pharmacy and say, give me some of this peptide. Or it's not like just going to Peptide Sciences or some other, provider that produces what they call research grade peptides. They're not even clinical grade. And you're using them with a very high risk because they're not produced in a CGMP from a clinical standpoint, clinical grade product. So what we have, our manufacturer is CGMP, FDA audited, and is producing a clinical grade product that's individualized to the patient. We also do that for augmented peptides. So like in other words, we took off the shelf peptides and we built scaffolding into them to make them much more vibrant and enhance their function. So now we have peptides that have what we call cell signaling scaffolding that allows for a little bit of the dosing goes a long way with less side effects and better targetability, because they're not just naked backbone structures. They're actually scaffold structures with cell signaling attachments to them, right? So we do that for the augmented peptides. And for augmented peptides, you don't need to have testing done. You can just know your indications if you got longevity patients or performance patients, or you just want to, you want to just work to, strengthen somebody's health. They're generally healthy. You want to have some kind of a prevention strategy, but these patients are not, super ill, right? They're just, you're just trying to stimulate vibrance for them. And there's a way of combining like augmenteds with personalized too, like in longevity. In your longevity, you get your personalized, but then you say, I really want to create more enhancement of mitochondria. So what can I use as an augmented peptide to assist that strengthening process, and work along with the personalized longevity peptides. So the thing is, we've got personalized peptides for active disease and cancer, all active diseases that I mentioned, autoimmune, neurodegenerative, diabetes, those type of things, Lyme disease, chronic infections, or whatever. And then you've got prevention for people that may, for instance, I had cancer 10 years ago, and I want to make sure that I'm doing stuff proactively to prevent it. We've designed prevention designs. And then we have the longevity personalized peptides that are specifically to the aging hallmark interface, and interventions there for potentiating, mitigating, regulating. And then we have the augmented peptides that are used in resilience and regenerative longevity for performance-based approaches, right? So we have that. So that's the thing. So we're not just going to a compounding pharmacy to get something compounded. It's manufactured direct for the patient on the personalization side. And even the augmented peptides are manufactured direct because they're very highly specialized structures, right? And it goes through that chain of custody. And we do that because it stays within a compliance. And then our physicians are also under an IRB. So in other words, they have their own IRBs constructed because they have the ability to write into the IRB, which we equip our doctors to be sure that they have an IRB structure to actually allow for safety of use in human subjects, right? And so that's another layer of compliance protection too for the practitioners as well. So the process doesn't take very long when the personalization. It's usually on average, about 8 weeks from sample submission to the output engineering to get the peptides. In more complicated cases, it might take up to 10 weeks because of the fact that we're waiting on tumor biopsy or whatever, right? But on an average, it's about 7 to 8 weeks usually. And then the longevity peptides are less than that. It's a lot of times we can get the longevity peptides in 7 weeks, and then the augmented peptides you can get in as little as 4 weeks. So.
And can you work with anyone anywhere in the world, or do they have to be in the US?
No, we actually are working with overseas, where our logistics for our partners, where we're just working out now, where we can ship, to other places. We'd have to know where it's going, of course, ahead of time. And the way our peptide manufacturer works is, they understand the whole dynamic of things getting caught up in customs and things like that. So we work with those kind of issues, and we'll let you know ahead of time. Is this a location that we can work in, or is it more complications in this area? Maybe you would have them go to a different area where there's less resistance, But the thing is that most of the time we're finding some cooperative movement in countries with maybe some twists and turns, And if that's the case, then we'll direct you as to what countries are more friendly for this, right? So.
Amazing. Well, for people listening, there's obviously a whole world opened up of potential where it comes to things, especially when you're diagnosed with something scary like cancer. But, end-stage kidney disease, the whole fibrosis thing we spoke about, if you can just upregulate part of the healthy tissue and get that crosstalk going there. Dr. Grinberg, is there anything you wanted to add before we wind this up?
Yes, so a few more things. So like John mentioned, some patients are very sick and they approach me and they ask me what they can do during these 8 weeks while the therapeutics manufactured by Neo7. So recently we had a meeting with a few more companies that provide advanced treatments for advanced cancers. One approach is using patients' proliferated NK cells that definitely can slow down most cancers, but has certain limitations compared to system-wide approach by Neo7. But in terms of helping patients to live long enough to reap benefits of Neo7, treatments proliferated and transfused back to patient NK cells frequently help a lot. There are companies that also work on creating individualized cancer vaccines that also can slow down the process. But again, Neo7 has most, I would say, all-inclusive and system-wide approach, and it would be great if people could live long enough to be able to benefit from the therapeutics developed by Neo7 for their individual treatment, as I see greatest potential in this approach. And it's interesting, over the time I'm working with Neo7, I also have friends, scientists, prominent immunologists, people in US Cancer Association. I put them in touch with Neo7, and their scientific opinion, they keep Neo7 platform in high regard, and they feel it's scientifically very sound and promising. And finally, another good news is we trying to develop some legal movement or getting petitions to advocate for patients being able to determine where the insurance benefits go to conventional medicine or to individualized, customized medicine location sites like Neo7. So to increase the freedom of choice for the patients to decide, particularly when they deal with such conditions where conventional medicine agrees that has little to offer.
Well, that's some very exciting stuff coming down the pipeline, and I feel like this is the beginning of how, hopefully, if Big Pharma doesn't get in the way, moves into how treatments should be, where we can have these very highly individualized tests. We can customize based off genome expression and actually imagine too, if everyone turned 20, had this done and started to go on individualized peptides to prevent disease. I mean, really the future looks very bright when we've got these type of technologies. So I want to thank you both for joining me today and, paving the way for what I do hope is the future of medicine, especially when it comes to scary diseases like cancer, chronic kidney disease, autoimmune diseases and things like that, and let people understand that there are greater choices out there, and their medicine is slowly moving towards this very highly individualized treatment and using very simple things, peptides. Got lots of podcasts on there with Dr. Camp and Dr. Grimberg, and there's lots of different people talking about them in the longevity world, but really therapeutics with very little side effects, and we're just upregulating and enhancing the way that the body communicates. So it's very exciting. So thank you both. I'm going to put Neo7's website in the show notes. I'll put a link to Dr. Grimberg if you want to work with a doctor who's very well versed in using longevity medicine peptides. He's in San Francisco in the US over there. And thank you both. Remember to hit like. That means you'll get notified anytime we put up new content. If you want to hear more, if you've got specific questions for these two, please let me know and I can pass it on below. If you want to know more about what we do, head to www.kidneycoach.com. Thank you for being part of our community. John and Dr. Grimberg, thank you both for your time. I highly appreciate you and what you're doing in the world. It is very exciting. Thanks all. Bye.
Thank you. Bye, Fiona. Bye, Dr. Grimberg.
Bye, John. Bye, Fiona.